So if this is a glutamatergic neuron, right, that's going to synthesize glutamate, the most common excitatory neurotransmitter in the brain, and release it upon the arrival of an action potential, well, it's going to be making contact with receptors that are localized in the postsynaptic membranes of the next neuron. I mentioned it's excitatory, right? It's the primary excitatory neurotransmitter in the CNS. So that means it'll bind certainly to a set of ionotropic receptors. And these ionotropic receptors are going to be excitatory, right? Which means that they've got to open channels, within their structures that can allow for depolarizing currents to pass through. So that means they open channels to sodium, right? And we're going to see also sodium, in some cases sodium and calcium. And so again, there are also metabotropic glutamate receptors that we're not going to talk about as much, but the ionotropic receptors are particularly important for, we're going to see normal excitatory transmission at synapses, normal sort of depolarizing transmission, and also for learning and memory because of the action of a particular kind of postsynaptic glutamate receptor, ionotropic glutamate receptor, called the Nmda receptor. Now, there's a number of distinct classes or subtypes of receptor that glutamate can bind postsynaptically and open channels with. There are two subtypes. One's called the kainate subtype of glutamate receptors, so the kainate glutamate receptors. The other one is called the Ampa glutamate receptors. And again, those two they're going to have very similar sorts of action, but they're distinguished because kainate is a chemical, right, that will only act as an agonist on the kainate subtype of glutamate receptor, while Ampa is another chemical that will act as an agonist on a second subset of glutamate receptor that we call the Ampa receptors. Remember, glutamate will act at kainate and Ampa, and we're going to see also these Nmda receptors. But kainate and Ampa receptors have a similar kind of response to the binding of glutamate. Glutamate will bind, and they will open up a channel, a hole, right? They're ionotropic, of course, right? They're ligand-gated ion channels, right? They'll open up a hole that will allow for sodium to pass through. And remember, postsynaptically, we're assuming that neuron is at its resting potential, which means minus 65 on the inside, which means that both the concentration gradient force, because sodium, remember, is stored in high concentration outside the membrane in a neuron at rest, and the electrical force, because sodium is a positively charged ion, and the inside is at minus 65. So both those forces will drive the sodium through the open Ampa and kainate channels, or receptors, and will depolarize, the cell towards, the threshold necessary for an action potential. So that's the mechanism there. Now, there's another type of receptor that we'll talk about next, a glutamate receptor. It's an ionotropic receptor, and it's known as the Nmda receptor. And this is a very specific type that's most definitely involved in aspects of learning and memory.